Let's start.
Cellular is circulatory too?
I'm sorry?
Cellular is circulatory too?
No, we're talking about what's happening inside the cells now.
Never mind.
Yeah. Okay, so now let's talk about B cells, B lymphocytes. We have about 100 million different types of B cells. What's different about them? Well, the receptors that they have on the surface. We'll talk about how we go about making all these different types later. But so you have a million, 100 million or so different B cells. And they are either circulating in the bloodstream, or as I've mentioned already, many of them are going to be, in the lymphatic system, in the lymph nodes and stuff. And really, there is where they get exposed to antigens and other things. So you have these B cells all doing their job, circulating and things. And one of them is going to come in contact with the right antigen. It's going to bind to it. This antigen doesn't fit here, but it fits here. This is called clonal selection. And clonal selection happens when the right B cell binds to the right antigen. And as you might expect, that B cell then replicates itself, with that antigen. So more of these can also bind to it, and it continues the process of replicating. And this is called clonal expansion. And so now you've got many, many B cells that fit that antigen. Okay? Of the 100 million B cells that you have circulating, that one B cell bound to that one antigen, had the right combination, the right surface protein, in binding to it, started to proliferate, clonal expansion, and then started to differentiate. Some of these cells became what are known as plasma cells. And plasma cells release antibodies. So now you've got all these plasma cells releasing all kinds of antibodies, flooding the bloodstream with antibodies, flooding the lymph with antibodies. And the antibodies then, we're talking lots and lots of antibodies, bind to all of the antigens that are out there. So now you've got this antibody floating around, and it's binding to this antigen, and it's over here, and it's binding to this antigen. So now the antigens are being bound by the antibodies, and so our macrophages and our neutrophils can recognize these guys and devour them. By binding these antigens with the antibodies, they get removed from the system. They can't replicate, and so eventually a macrophage will come and take care of them, devour them. Some of these B cells will become what we call memory B cells. And memory B cells do just that. They recognize this antigen, and if they see it again, they can respond immediately with antibody production. See, what I haven't told you yet is this clonal expansion that goes on needs input. It doesn't happen automatically. The B cell that binds to this antigen, it needs information. It's not enough that it recognizes that antigen. What it has to do is now take that antigen and present it to a T cell. So, before this has happened, the B cell has to present that antigen to the T cell. What it does is the B cell will internalize the antigen, break it up into lots of different parts, and then present those different parts on the surface. So those are three different parts to the antigen that it bound. So it picked up this antigen, the B cell did, so this is my B cell here. And it picked it up because it recognized it. It had a surface protein that recognized the antigen, internalized it, so brought it inside the cell, and then broke it up into smaller pieces. And then took those smaller pieces and presented them on the surface. So now the B cell is presenting these molecules to T cells. And so a T cell comes along here now. And a T cell, also highly specific, will only bind to a specific molecule that it's being presented to, comes in contact with this B cell. And it doesn't recognize this cell that it's being presented, but it turns out that it does recognize this one as it's being presented. Let me write this as a T cell here. So, in recognizing this epitope that's being presented, this antigen that's being presented, the T cell then can release cytokines. And of course, our B cell has on its surface receptors specific for these cytokines. So those cytokines bind to the B cell surface, and that initiates the clonal expansion that we see here. So the B cells will only undergo clonal expansion if the T cells tell them it's okay. The T cells have to interpret what is being presented to them. And then if it is something that needs to be responded to, something that has been recognized as bad, then they'll release cytokines and clonal expansion will take place. If the T cell doesn't recognize any of these, then that'll be ignored and clonal expansion won't take place.
It only has to recognize one of them
Because there is likely other T cells out there. So there's probably another T cell out there with a receptor that might recognize this one. Okay? So there's likely a couple of T cells that might recognize the different receptors that are out there. But our story's not quite done there. How does our T cell know that this is a receptor that it needs to be aware of? Well, remember back here in our inflammatory response, in our inflammation, we had all those bacterial products that we were being exposed to and stuff. In and among those cells are dendritic cells. Now, dendritic cells are drawn with a fairly odd shape to them, kind of a floral shape to them. And their main function, main role, is really to pick up different antigens. So it's basically engulfed these different antigens from the inflammatory location. And now it makes its way into the lymphatic system. Okay? And as it makes its way into the lymphatic system, so we write this as, we have this as a dendritic cell. This dendritic cell is going to migrate into the lymphatic system. Where it will come in contact with T cells, helper T cells. And so on the surface of our dendritic cell, it will present the different antigens that it has encountered. Okay? Well, we have a T cell now that's in the lymphatic system, but it's not been presented ever with an antigen. So we call it a... Is that how you spell naive? I think so. I hope so. Take my word for it. So we have a naive T cell. This T cell has not been exposed to any antigens, but it still has on its surface receptor specific to an antigen. So it's just waiting for somebody to come and expose it to an antigen. Well, it just happens that this dendritic cell has picked up an antigen that is going to fit into this T cell. Oop. Now recognize, just like the B cells, you've got 100 million different T cells. So you've got a lot of different T cells, and one of them hopefully is going to match the antigen that's being presented. So this T cell here, identify it as a T cell. And specifically, it's a helper T cell. This T cell recognizes the antigen that this dendritic cell is presenting to it. But that's not enough. It needs more than just to recognize the antigen. See, additionally, located on the surface of our dendritic cell are toll-like receptors. Just like the toll-like receptors we find on the macrophages. And these toll-like receptors bind to peptoglycan, bind to lipopolysaccharide, bind to flagellin. Okay, so if this antigen that is being presented to the T cell happens to come from a bacterial cell, and we would know it's coming from a bacterial cell because these toll-like receptors have been bound, then the dendritic cell releases a chemical called a co-stimulatory molecule. A co-stimulatory molecule serves like a flashing red light, okay, telling this T cell that the antigen that you are now encountering and reading is coming from a bacterial cell. And so there's a red light, you need to pay attention to it, you need to recognize it, you need to respond accordingly. In the presence of a co-stimulatory molecule, the T cell then will become an effector T cell. An effector T cell can now go out into the circulatory system, be presented with an antigen from the B cell, and cause that B cell then to undergo clonal expansion. If the co-stimulatory molecule is not present during this presentation of the antigen, then the T cell will ignore that antigen, and it will not trigger an immune response. Questions?
So it needs to both recognize what the dendritic cell has picked up and have the co-stimulatory present in order to respond.
Exactly. So it isn't enough to just recognize the antigen. If the co-stimulatory molecule isn't being released, then the T cell is going to ignore the antigen. The co-stimulatory molecule serves to tell the T cell that it's a bacterial product that it's being presented. I cannot get this computer to work, no matter what I do. Okay, so a couple of things that we can talk about here. I've been talking about antigens and antibodies. Let's be clear on what they are. First off, an antigen, also known as an antibody generator. Okay? This is going to be anything that then generates the production of antibodies. It's usually recognized as foreign to the host. And when we talk about an antigen, there are discrete regions of the antigen called epitopes that are really is what is being recognized. So in our example of our bacteria here that's being internalized by the B cell and then presented, 3 different parts of that bacteria are presenting. Each one of those serves as an epitope. So you can take a bacteria, and this part of it might be a region that serves as an epitope. There might be molecules here that serve as an epitope. There might be something internal that serves as an epitope. So anything that is being presented, the region that is being recognized is the epitope. Anything that is being bound to, the region that is being recognized. Antibodies then, these things are glycoproteins, and we'll find them in a Y shape. There are 5 major classes of antibodies. I won't ask you to know the 5 classes. We may talk about a couple of them later on. The antibody is composed of 2 main active parts, if you will, functional parts.